Welcome to our cell culture laboratory. It includes analyses to examine the effects of compounds on cells by culturing mammalian cell lines in a sterile environment, ensuring reliable and safe experimental results. This is the Class II biosafety cabinet. All cell culture work is carried out inside this cabinet to ensure sterility. The system uses a constant flow of HEPA-filtered laminar air to remove dust, bacteria, and spores from the working spaces. The cells are maintained under sterile conditions with UV protection to prevent contamination and ensure reliable experimental outcomes. Here you can see the CO2 incubator, which provides the optimal conditions for cell growth. It keeps the temperature stable at 37 degrees, maintains high humidity, and ensures the correct balance of carbon dioxide and oxygen. This water bath warms culture media and reagents to 37 degrees, preventing sudden temperature changes. This is the inverted microscope, specially designed for observing live cells in culture flasks and plates. This is the refrigerated centrifuge, a laboratory instrument designed to separate biological materials, such as whole cells, from their surrounding solution. By applying high centrifugal force, it enables the isolation and purification of these components for further analysis. This is the standard light microscope, one of the most basic yet fundamental tools in biology. It allows us to examine stained slides and study the morphology of cells in detail. We can identify structural changes, count cell numbers, and observe different cellular features that are not visible with the naked eye. This is the autoclave, a device used to sterilize glassware, instruments, and solutions. Finally, we have the microplate reader, which enables high-throughput analysis of samples. It measures absorbance, fluorescence, or luminescence in 96-well plates. This is a cell imaging microscope, used for observing cells in detail, including their morphology, behavior, and dynamic processes such as wound healing. This is a research microscope with a digital imaging system, commonly used in education, clinical diagnostics, and biomedical research to observe cells, tissues, and fluorescence-labeled samples in high detail. It allows researchers to visualize specific cellular structures or molecules by tagging them with fluorescent dyes or proteins, providing insights into complex biological processes. In this section, we demonstrate how to properly thaw frozen cells. We start by retrieving a frozen vial of cells from liquid nitrogen storage at -196 degrees Celsius. The cells are rapidly thawed in a 37 degrees Celsius water bath until mostly dissolved. Once thawed, they are transferred into a falcon tube containing pre-warmed growth medium. Gentle pipetting ensures the cells are evenly suspended. Once the cells have been resuspended in the growth medium, they need to be transferred into appropriate culture vessels to support their proliferation. Proper labeling and handling at this stage are essential to ensure accurate tracking and reproducibility of experiments. Next, the cells are moved into a labeled flask, indicating the cell line, date, passage number, and researcher name. The cell suspension in the falcon tube was transferred to the flask using a sterile serological pipette. After a brief microscopic observation, the flask is placed in a 37 degrees Celsius incubator with 5% CO2 to allow the cells to grow. The cells are monitored regularly to assess their morphology and confluency. Medium changes are performed as needed to provide fresh nutrients and maintain optimal growth conditions. Any signs of contamination are carefully checked to ensure the integrity of the culture. Next, we show how to replace the culture medium to maintain healthy cell growth. First, we carefully observe the cells under the microscope to assess their health and confluence. Changing the medium is essential to provide fresh nutrients, remove metabolic waste, and support optimal growth. The old medium is gently discarded into a waste container. Fresh medium is added to the flask, ensuring it spreads evenly across the surface, so every cell receives sufficient nutrients. Care is taken to avoid disturbing the cells during the addition. The cells are gently observed under the microscope to confirm even distribution and attachment. Any clumps or bubbles are carefully dispersed to maintain optimal growth conditions. Finally, the flask is returned to the 37 degrees Celsius incubator with 5% CO2 for continued growth. In this section, we demonstrate cell subculturing. We start by observing the cells under the microscope. When they cover around 80% of the flask surface, they are considered confluent and ready for passage. Subculturing helps dilute the cells, giving them more space and nutrients to continue growing. In the biosafety cabinet, We remove the old medium and gently wash the cells with phosphate buffer solution to remove metabolic waste and dead cells. This step ensures a clean environment for the cells, promoting healthy growth and accurate experimental results. Care is taken to avoid dislodging adherent cells during the washing process. Adherent cells are cells that attach and grow firmly on the surface of a culture vessel, such as a flask or plate. Their adhesion is essential for normal cellular functions, including proliferation, signaling, and morphology. Next, we add trypsin EDTA to detach the cells from the flask surface and incubate for 2 to 3 minutes at 37 degrees Celsius. Trypsin EDTA is used to break down the proteins that mediate cell adhesion, allowing the cells to detach from the flask surface. The incubation at 37 degrees Celsius provides an optimal temperature for enzymatic activity. The duration of incubation may vary depending on the specific cell line, as some cells detach faster or more slowly than others. The enzyme is evenly distributed across the entire surface of the flask containing the cells. Cells detached from the surface by the enzyme can be observed moving under the microscope. Once detached, we check the cells under the microscope, and then neutralize the enzyme by adding fresh medium. After adding the medium, the cells are gently pipetted several times across the flask surface to ensure that all detached and remaining adherent cells are collected. Some cells may remain attached to the flask surface even after trypsin EDTA treatment. In such cases, a cell scraper can be used to gently dislodge these resistant cells. Care must be taken to avoid damaging the cells, while scraping to maintain their viability for further experiments. The resulting cell suspension is then carefully transferred into a falcon tube for subsequent centrifugation or further processing. The cell suspension is collected and centrifuged to form a pellet. The centrifuge used in cell culture is typically refrigerated to maintain a low temperature, which helps preserve cell viability and prevent stress or damage during the process. Cooling is particularly important for sensitive or primary cells that may be adversely affected by heat generated during centrifugation. Using a refrigerated centrifuge ensures that metabolic activity is minimized, reducing the risk of cell death or unwanted changes in cell behavior. Proper balancing of the tubes is also essential to achieve efficient pelleting and prevent mechanical damage. After removing the supernatant, the cells are resuspended. Their viability is checked with trypan blue under a light microscope, and the appropriate number of cells are seeded into new flasks with proper labeling. Finally, the flasks are returned to the incubator for continued growth. To ensure the pellet is evenly resuspended, it is important to gently disperse it manually and thoroughly pipette it with fresh culture medium. Another preferred step before seeding the cells into the flask is performing a cell count. For this purpose, a small volume of the cell suspension is mixed with an equal volume of trypan blue, and then loaded onto a TBAE or Neubauer chamber for viability assessment. Trypan blue penetrates the membranes of damaged cells, staining them blue, while viable cells appear transparent and bright. Cell viability is analyzed under a light microscope using the grid counting method. The Neubauer chamber contains a precisely defined grid that allows accurate calculation of cell concentration in a given volume. This standardized design ensures reproducibility and reliability in cell counting experiments. After centrifugation, depending on the pellet density or following cell counting, the desired concentration of cell suspension is prepared and distributed into several flasks containing growth medium. The surface of each flask is labeled with the cell line name, passage number, passage date, and researcher's name. The cells are then expanded and maintained in a healthy state in an incubator set at 37 degrees Celsius with 5% CO2 and controlled humidity. In this section, we demonstrate a cell incorporation experiment to observe changes in cell uptake over time. We start by passaging the cell line to make sure they are healthy and ready for the experiment. To seed the 24-well plate, we first count the cells. Before proceeding with this step, the cell passaging procedure described in the previous section is carried out. A small sample of the cell suspension is then mixed with trypan blue and placed on a TBAE chamber. Under the microscope, we count the viable cells to calculate how many cells to add to each well. Using the TBAE chamber, both cell viability and total cell number can be accurately determined. Once calculated, the cell suspension is carefully added to the wells, making sure the cells are evenly distributed. The plate is then placed in a 37 degrees Celsius incubator to allow the cells to attach and form a uniform layer, creating our experimental group. After 24 hours incubation, the cells are examined under the microscope to check their attachment and density. We remove the old medium and gently wash the wells with PBS. Washing the wells twice with PBS is important to remove residual medium and metabolic waste, which could interfere with subsequent steps. This process also helps eliminate dead or detached cells that may affect cell health and experimental accuracy. Ensuring a clean environment in the wells promotes optimal conditions for further treatments or assays. Depending on the study design, the experiment is carried out in triplicates or quintuplicates to ensure statistical reliability, as the number of replicates is important for the stability and reproducibility of the experiment. An incorporation assay is a type of experiment used to measure the uptake of specific molecules, such as nucleotides labeled substrates, or compounds tagged with isotopes like technetium, 99M, iodine, 131, and gallium, 68, into cells. This method provides valuable information about cellular metabolism, proliferation, and overall activity under different experimental conditions. The radiolabeled compound is then added to the cells. Before measuring the initial activity of the radiolabeled compound, the sample is prepared for detection using a cadmium telluride detector, with 3 replicates and a measurement duration of 5 seconds per replicate. Cadmium telluride (CDTE) is a semiconductor material commonly used in radiation detectors, due to its high efficiency in converting gamma or X-ray photons into electrical signals. A channel range is selected according to the radionuclide used, defining the wavelength range to be detected. Subsequently, the background of the medium is measured. This step is crucial for ensuring the accuracy of the measurement results and verifying that the detector is functioning correctly. Immediately, we measure the initial activity in selected wells. After measuring the initial activity, the radiolabeled medium in the wells is removed at the predetermined time points, and the wells are washed with PBS. Measurements are then performed using the cadmium telluride detector within the previously defined channel range to analyze the time-dependent interaction between the cells and the radiolabeled compound.